Today we're going to continue our lesson on cellular energy, and we will be focusing on cellular respiration. We need to make sure that we understand a few of our vocab words, including the word metabolism. Metabolism is a word that we commonly use in our everyday language, but in scientific terms in biology, metabolism is the combination of the chemical reactions of an organism that build up or break down materials. This does tie in part to when we talk about organisms getting their food, the energy they need from their food. Remember, we get energy from our food in the form of carbohydrates, fats, and proteins. When we talk about our food contains chemical energy, and the energy is released from that food when those chemical bonds are broken. Our cells use energy that is in those chemical bonds to make ATP. ATP stands for adenosine triphosphate. ATP is a molecule that the cells use to store and release energy. ATP is the energy currency of the cell. It's the basic source of energy used by all cells. An ATP molecule is made up of adenine, ribose, and a phosphate group. When we talk about our cellular respiration, we need to understand there is an aerobic and anaerobic processes. These processes are involved in cellular respiration, and an aerobic process requires oxygen, and they take place when oxygen is available. This takes place in all eukaryotic multicellular organisms. Aerobic involves oxygen. Anaerobic does not require oxygen. So anaerobic is the taking place in the absence of oxygen. And this takes place in many of our singular celled prokaryotes. Cellular respiration is defined as the series of processes that release energy from food in the presence of oxygen. Think about this. We consume food. We also breathe in oxygen. Your cellular respiration is a series of controlled actions that gradually releases the energy that is stored in the food. Cells transfer the energy that is stored in food to that usable energy of ATP. Your chemical equation for cellular respiration is C6H12O6, or glucose, plus 6 molecules of oxygen, which yields 6 CO2 molecules, or carbon dioxide molecules, 6 water molecules, and ATP. This chemical equation should look familiar because it is the reverse of what is happening in photosynthesis. The carbohydrate glucose reacts in the presence of oxygen to produce a carbon dioxide gas, water, and ATP. When we exhale, our waste products of cellular respiration are carbon dioxide and water, and we release those as we exhale. The energy is what your cells need in order to carry on all their processes. Most of the reactions that take place in cellular respiration occur within the mitochondria. Remember, we refer to the mitochondria as the powerhouse of the cell, so it's where the energy is generated. The mitochondria convert chemical energy in the food into that usable ATP that cells use to power all of the activities in the cell. This is a general overview of cellular respiration. There are 3 steps of the cellular respiration process. There's glycolysis, the Krebs cycle, and the electron transport chain. You can see here what is produced from each one of these steps of cellular respiration. Glycolysis produces 2 ATP. The Krebs cycle produces the waste product CO2. And in the electron transport chain, this is where the bulk of your ATP is produced. There are 34 ATP produced from the electron transport chain, and this is also where the waste product H2O is produced. You're now going to watch a short video that is an introduction to cellular respiration.
The cell needs to be able to harvest energy from glucose to get the usable energy, ATP. Whether oxygen is present or not, cells can always do glycolysis, which occurs in the fluid cytosol of the cell. No oxygen is needed for this. During glycolysis, the 6-carbon glucose molecule is broken in half and rearranged to make 2 pyruvic acid molecules. The process requires 2 ATP to get started, but ultimately it produces 4 ATP from ADP and phosphate, and it produces 2 NADH from NAD+. So overall, glycolysis makes 2 pyruvic acids, 2 ATP, and 2 NADH. From here, there are 2 possible pathways that could be taken. Cellular respiration always starts the same way, with glycolysis. From there, the 2 pathways are determined by the presence of oxygen. If there is no oxygen available, anaerobic respiration will occur. But if oxygen is available, aerobic respiration will occur in the mitochondria. Let's look at anaerobic respiration first. The anaerobic pathway has another name, fermentation. Fermentation is an anaerobic process that converts sugars to acids or gases or alcohols. It includes the glycolysis step in it. Fermentation replenishes the supply of NAD+ so that glycolysis can continue, creating 2 ATP from each glucose molecule. There are 2 types of fermentation. There's alcoholic fermentation and lactic acid fermentation. In lactic acid fermentation, pyruvic acid is converted to a chemical called lactic acid. The process changes NADH back to NAD+, so that glycolysis can continue and produce the 2 net ATP. Fermentation itself doesn't create any ATP, it's just keeping the glycolysis running. Our muscles use lactic acid fermentation when there's not enough oxygen to do aerobic respiration. Your muscles will feel cramped and painful, but this pain will go away as soon as you get enough oxygen back to your muscles so that you can go back into aerobic respiration. Other organisms use a different pathway and create an alcohol and carbon dioxide instead of lactic acid from pyruvic acid. Again, they still make NAD+ for glycolysis to use, but the pathway is a little different. Yeast will undergo alcoholic fermentation, and we use it to our advantage when making bread or beer. The alcohol and bubbles in beer are made by the yeast through alcoholic fermentation. When bread dough is left to rise, the rising comes from the production of carbon dioxide bubbles in fermentation. The alcohol in the bread cooks off in the baking process. Now, anaerobic respiration is not actually very good at making ATP. It relies on just the 2 ATP that can be made from glycolysis. But if oxygen is present, aerobic respiration will occur. And it's an ATP-producing machine. Aerobic respiration takes place in the mitochondria of eukaryotic cells and in the cytosol of prokaryotic cells. After glycolysis occurs in the cytosol and pyruvic acid is attached to coenzyme A, there are 2 major parts. The Krebs cycle, also known as the citric acid cycle, and the electron transport chain, which is paired with chemiosmosis. This last step is where oxygen is used. During the Krebs cycle, 2 ATP and the gas carbon dioxide are produced. The carbon dioxide will eventually diffuse out of the cell. This is why we breathe out carbon dioxide. The electron transport chain and chemiosmosis take place in the folded inner membrane of the mitochondria called the cristae. In the end, aerobic respiration can make up to 38 total ATP, theoretically, but usually it's a bit less than that, more like 30 to 32 ATP. So the final reaction is glucose plus oxygen yields 6 CO2 and 6 water, plus energy in the form of ATP. And lots of it.
Glycolysis is the first step in cellular respiration. It is an anaerobic process. In glycolysis, glucose is broken down. This takes place within the cytoplasm. And what happens is, as the bonds of glucose are broken down and they get rearranged, energy is released. Remember, that's an important idea to think about, because it goes back to those laws we reviewed at the beginning of this, that energy can't be created or destroyed. It just gets used in a different manner. One molecule of glucose is broken down into 2 molecules of pyruvic acid, and that moves on to the Krebs cycle. 4 electrons are passed to the carrier NAD, which produces NADH, and that's the carrier that the electrons to the electron transport chain. 2 ATP molecules are invested to get the process of glycolysis moving, because again, this is a process, so it does require energy as well. 4 ATP molecules are produced, so there is a gain of 2 ATP molecules of glucose per molecule of glucose made in glycolysis. So for every glucose, 2 ATP are produced in glycolysis. About 90% of the energy available in one glucose molecule is still unused at this point, and it's locked up in the chemical bonds of the pyruvic acid. The pyruvic acid from glycolysis is used to make carbon dioxide, ATP, and the electron carrier molecules in a series of energy extracting reactions in the Krebs cycle. So we move from glycolysis into the Krebs cycle. This is also called the citric acid cycle, because the first molecule formed is a citrate, and it's a type of citric acid. The Krebs cycle is aerobic, meaning that it requires oxygen in order for it to take place. It takes place within the matrix of the mitochondria. This is the innermost compartments of the mitochondria. The steps of the Krebs cycle are: step 1, acetyl-CoA combines with OAA to produce that citric acid. Then citric acid goes through a series of reactions to release the energy that's captured in the molecules of NADH, ATP, and FADH2. Carbon dioxide is released as a waste product. The OAA is regenerated and used for the next turn through the cycle. 2 turns are needed because glycolysis produces 2 pyruvic acid molecules when it splits the glucose. At the conclusion of the Krebs cycle, carbon atoms from the acetyl-CoA combined with oxygen to form 4 molecules of carbon dioxide. The energy from the chemical bonds is stored in the energy carrier molecules. So at the end of the Krebs cycle, we have 6 molecules of NADH, 2 molecules of FADH2, and 2 molecules of ATP. You'll now watch a short video about ATP synthesis.
All life on Earth depends on this tiny energetic molecule, adenosine triphosphate, or ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA. Generating ATP for your living cells are mitochondria, electrochemical batteries that convert energy from the food you eat and oxygen from the air you breathe into ATP. So where does your ATP come from? Deep inside your mitochondria, rows of molecular motors generate ATP, the molecule essential to all life on Earth. Enzymes bring together reactants to form a chemical bond, converting mechanical energy into chemical energy. A ring of enzymes work in step, creating 3 molecules of ATP with each cycle. Inside the molecular motor, a rotating axle powers the sequence. The axle is attached to a rotary molecular motor, moved by the force of protons pushing from the other side of the membrane. A difference in proton concentration propels the molecular mechanism. Synthesis of ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA.
The electron transport chain. This is the final stage of the aerobic cellular respiration, and it is a series of proteins and molecules. It is within the folded inner mitochondrial membrane called the cristae. It uses high energy electrons from glycolysis and the Krebs cycle to convert ADP into ATP. Energy is released in these reactions and captured as a proton gradient, which is then used to make ATP. Together, the electron transport chain and chemosis make up oxidative phosphorylation, and this occurs in steps. First, the protein pumps create an electrochemical gradient. The ATP synthesis uses diffusion of protons to synthesize the ATP. Oxygen accepts the electrons and protons to form water. Keep in mind the 2 waste products from our cellular respiration are carbon dioxide, water, and then we also have the ATP being produced. The inner mitochondrial membrane contains enzymes called ATP synases that rotate when hydrogen ions move through them across the cell membrane. With each rotation, it attaches to the phosphate group to ADP to generate ATP. The electron transport chain yields about 30 to 32 molecules of ATP per molecule of glucose. The end result. So together, glycolysis, the Krebs cycle, and the electron transport chain release about 36 molecules of ATP per molecule of glucose. The 36 ATP molecules generated represent about 36% of the total energy in glucose. The remaining 64% is released as heat. So you will now watch another short video about our cellular respiration process.
Are you a morning person? One of us is, and one of us is definitely not. Mainly because when I wake up in the morning, it just takes a while for me to get my energy back. It takes a lot of time and coffee for that to happen for me. Cells really don't have that luxury. They are busy performing cell processes all the time. Active transport of many substances needed for their survival, for example. And the energy currency they need, specifically, is ATP. ATP stands for adenosine triphosphate. It's a type of nucleic acid, actually, and it is action-packed with 3 phosphates. We have a video all about ATP and how it works as an energy currency. So where am I going with this? Well, cells have to make this ATP. It doesn't really matter what kind of cell you are. Prokaryote or eukaryote, you have to make ATP. The process for making that ATP can be different, however, depending on that type of cell. One way is called aerobic cellular respiration. Lots of organisms can do aerobic cellular respiration, but this video is specifically going to go into aerobic cellular respiration in eukaryotic cells. That means this video is talking about the process within cells that have membrane-bound organelles, such as a nucleus and mitochondria. Eukaryotic cells include the cells you'd find in protists, fungi, animals, and plants. The mitochondria, which can be found in most eukaryotic cells, are going to be kind of a big deal in this aerobic cellular respiration because some of the process occurs in the mitochondria. So let's get started. Remember, the major goal for any organism performing this is to make ATP. Okay, here's the overall look at the equation for aerobic cellular respiration. Remember that reactants, inputs, are on the left side of the arrow, and products, outputs, are on the right side of the arrow. This equation, by the way, looks remarkably similar to photosynthesis. Look at how the reactants and products are on different sides. While that doesn't mean they're simply opposites, it does show that they have substances in common. In photosynthesis, organisms make glucose. Notice how glucose is a product. But in cellular respiration, organisms break the glucose down to make ATP. Fun fact, you know how when a bean seed first germinates in the ground, it isn't able to do photosynthesis yet? Yeah, the germinating bean is relying on glucose that it has stored, and it's doing cellular respiration to break it down to make ATP so it can grow. Of course, once it starts to mature and develop leaves, it can do photosynthesis. It will be able to do both photosynthesis and cellular respiration. Plants make glucose in photosynthesis, and they break it down in cellular respiration. They can do both. But if you aren't photosynthetic, such as a human or an amoeba, you have to find a food source to get your glucose. You need glucose to get this process started, and we're going to assume that we're starting with one glucose molecule. Step 1, glycolysis. This step takes place in the cytoplasm, and this step does not require oxygen. It's considered anaerobic. In glycolysis, glucose, the sugar from the equation, is converted into a more usable form called pyruvate. Glycolysis usually takes a little ATP itself to start up. The net yield from this step is approximately 2 pyruvate and 2 ATP molecules, and 2 NADH. What is NADH? NADH is a coenzyme, and it has the ability to transfer electrons, which will be very useful in making even more ATP later on. We'll get to that in a minute. Now, an intermediate step occurs. The 2 pyruvate are transported by active transport into the mitochondria, specifically the mitochondrial matrix. In the mitochondria, pyruvate is oxidized. The 2 pyruvate are converted to 2-acetyl-CoA, which will be used by the next step. Carbon dioxide is released and 2 NADH are produced. Step 2, the Krebs cycle, also called the citric acid cycle, still in the mitochondrial matrix. The citric acid cycle is considered an aerobic process. While the cycle doesn't directly consume oxygen, some of the events in the cycle need oxygen to continue. To learn more about this intricate cycle where the 2-acetyl-CoA will enter, check out the further reading suggestions in the video details. Carbon dioxide is released. We also produce 2 ATP, 6 NADH, and 2 FADH. FADH is also a coenzyme, like NADH, and it will also assist in transferring electrons to make even more ATP. Step 3, the electron transport chain and chemiosmosis. This is just a beautiful thing, really. In eukaryotic cells, this is still in the mitochondria, and to be more specific, it's involving the inner mitochondrial membrane. We do require oxygen for this aerobic step. This is a very complex process, and we are greatly simplifying it by saying that electrons are transferred from the NADH and FADH to protein complexes in electron carriers. The electrons are used to generate a proton gradient, as protons are pumped across to the intermembrane space. All these protons being pumped out into this intermembrane space generates an electrical and chemical gradient. The thing is, if you remember from our cell transport video, ions, like the H+, don't easily travel across membranes directly without something to travel through. The protons can travel through an amazing enzyme called ATP synthase. If I could be any enzyme, I'd be ATP synthase because it has the ability to make ATP by adding a phosphate to ADP. ADP is a precursor to ATP. ADP has two phosphates, but if it obtains a third phosphate, it becomes ATP. So, in chemiosmosis, the protons travel down their electrochemical gradient through a portion of the ATP synthase, powering it to make ATP, the ultimate goal. Oxygen is the final acceptor of the electrons. When oxygen combines with two hydrogens, you get H2O, water. If you remember from our equation, water is a listed product. Now, the electron transport chain in chemiosmosis step makes a lot more ATP compared to the other two previous steps. How much? So I've noticed in my years of teaching and the assortment of textbooks I've collected over the years, there are some different numbers in cellular respiration charts. In fact, you can even see them change a bit in different editions of the same book. And it's made me want to emphasize that it's important to not just memorize a number, because it really is more of a range. I want to focus less on the number of ATP made per glucose molecule because it depends on a lot of variables. One variable is the gradient we were talking about, how many protons were pumped across the mitochondrial membrane. You can see more variables discussed in the factual references, and those references have estimates ranging from 26 to 34 molecules of ATP per glucose molecule in the electron transport chain in chemiosmosis step alone. Then, if you add the other two steps, Krebs, otherwise known as the citric acid cycle, and glycolysis, you could estimate a range of anywhere between 30 to 38 net ATP molecules total per molecule of glucose. Again, to emphasize a range. Now, this was just one way of creating ATP. But like we had said at the beginning, all cells have to make ATP, but the way they do it can differ. If there's no oxygen available, some cells have the ability to perform a process known as fermentation. It's not as efficient, but it can still make ATP when there isn't oxygen. We have a video about that. We can't emphasize enough how important the process of making ATP is for cells. For example, cyanide, which is found in some rat poisons, can block a step in the electron transport chain, which would block ATP production. A poison that prevents ATP from being produced can be deadly. With the important role that mitochondria have in ATP production, there is also a demand for increased research on mitochondrial diseases. We are confident that the understanding of how to treat these diseases will continue to improve as more people like you ask questions. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So, cellular respiration and photosynthesis. The relationship between cellular respiration and photosynthesis is what enables life on our planet to survive. The products of one process are reactants of the other process. Make sure you copy these two equations down, and you should see that these two equations are opposite of each other. In cellular respirations, glucose and oxygen come together and yield carbon dioxide and water, and in photosynthesis, carbon dioxide and water come together and yield glucose and oxygen. The process of respiration and photosynthesis cycle matter and energy in the biosphere. Photosynthesis makes the glucose that is used in cellular respiration to make ATP for plants. Water is broken down to form oxygen in photosynthesis, while oxygen is combined to form water in cellular respiration. Photosynthesis requires carbon dioxide and releases oxygen, while cellular respiration requires oxygen and releases carbon dioxide. It is important to remember that plants also undergo cellular respiration. The Glucose that is produced in photosynthesis then moves to the mitochondria, where the mitochondria use that to provide the energy that the plant needs. Fermentation. Sometimes oxygen is not available and organisms still need to undergo cellular respiration for energy. Fermentation is the anaerobic process of producing ATP from food in the absence of oxygen. Glycolysis is followed by a pathway of fermentation. This produces 2 ATP. And remember, that's much less ATP than aerobic respiration. This takes place in the cytoplasm of the cell. There are 2 main types of fermentation. There is alcoholic fermentation and lactic acid fermentation. In plants and yeast, the pyruvate from the glycolysis is converted into ethanol, alcohol, and carbon dioxide. In animals, some microorganisms, the pyruvate from glycolysis is converted into lactic acid. Both types of fermentation regenerate NAD so that the glycolysis can continue. You'll now watch a short video about the microorganisms in your food.
Where does bread get its fluffiness? Swiss cheese its holes? And what makes vinegar so sour? These foods may taste completely different, but all of these phenomena come from tiny organisms chowing down on sugar and belching up some culinary byproducts. Let's start with yeast. Yeast are single-celled fungi used to make bread, beer, and wine, among other products. Yeast break down carbohydrates like sugar to get energy and the molecules they need to function. They have 2 different ways to do this: the oxygen-dependent or aerobic pathway and the oxygen-independent anaerobic pathway, which is also called fermentation. When you bake bread, yeast can use both pathways, but they normally prefer to start with the anaerobic process of fermentation. In this process, ethanol is produced in addition to CO2. No, bread isn't alcoholic. Small amounts of alcohol that are secreted evaporate during baking. In the aerobic or oxygen-dependent pathway, the yeast consumes some of the sugar and produce carbon dioxide gas, or CO2, and water. In both processes, the CO2 accumulates and creates tiny bubbles. These bubbles get trapped by gluten and create a sponge-like structure that gives the bread its soft texture. Wine also relies on yeast, but a winemaking setup keeps the oxygen levels low so that yeast consumes sugar using fermentation, the anaerobic pathway. The process often starts with wild yeasts already hanging out on the grapes. But to get consistent results, most winemakers also add carefully selected strains of yeast that can tolerate high levels of alcohol. The yeast consume the sugar in the grape juice, and as the sugar level drops, the alcohol level rises. This doesn't necessarily mean that sweeter wines have less alcohol. Different types of grapes start with different amounts of sugar, and sugar can also be added. What happens to the carbon dioxide? It just bubbles away through a vent. In carbonated alcoholic beverages like champagne and beer, sealed containers are used in primary or secondary fermentation to keep the carbon dioxide in the bottle. Wine also introduces us to our second type of food-producing microorganism, bacteria. A special strain of bacteria turns a tart compound in grape juice into softer tasting ones that are responsible for some of the flavors in red wines and Chardonnays. Another type of bacteria, called acetic acid bacteria, isn't so desirable in wine, but they have their function too. If there's oxygen around, these bacteria convert the ethanol in wine into, well, acetic acid. Let this process continue, and you'll eventually get vinegar. Bacteria are the key for cheese, too. To make cheese, milk is inoculated with bacteria. The bacteria gobble up the lactose, a kind of sugar, and produce lactic acid along with many other chemicals. As the milk gets more and more acidic, its proteins start to aggregate and curdle. That's why spoiled milk is clumpy. Cheesemakers usually add an enzyme called rennet, naturally found inside of cows, goats, and some other mammals, to help this process along. Eventually, those little curdles turn into bigger curds, which are pressed to squeeze out the water and create a firm cheese. Different strains of bacteria make different kinds of cheese. For example, a species of bacteria that emits carbon dioxide is what gives Swiss cheese its characteristic holes. Some cheeses, Brie and Camembert, use another kind of microorganism too, mold. So your kitchen functions as a sort of biotechnology lab, manned by microorganisms that culture your cuisine. Yogurt, soy sauce, sour cream, sauerkraut, kefir, kimchi, kombucha, cheddar, challah, pita, and naan. But maybe not all at the same dinner.
Alcoholic fermentation. Microorganisms, including yeast, undergo alcoholic fermentation. It is pyruvic acid plus NADH, which yields alcohol plus CO2 plus NAD. This produces alcohol and carbon dioxide. It's used to produce alcoholic beverages and baked goods. Yeast production yeast will produce carbon dioxide bubbles in bread and dough, creating the little air holes that you see inside it, and it gives it that fluffy texture. Lactic acid fermentation. Most organisms, including humans, carry out the lactic acid fermentation when oxygen is not available. This would be pyruvic acid plus NADH, which yields lactic acid plus NAD. Lactic acid is used to provide a variety of foods such as cheese, yogurt, and pickles. It gives foods their sour taste. In most organisms, including humans, when they carry out lactic acid fermentation, it's for quick bursts of energy. The body uses ATP stored in the muscle cells, and ATP made by the lactic acid fermentation. The lactic acid byproduct of these reactions builds up in the muscles and causes that sore burning sensation that you get. For exercise longer than about 90 seconds, aerobic cellular respiration is the only way to continue generating an ATP supply. So you will now watch a short Amoeba Sisters video about a review of fermentation.
When I was a kid, swimming was one of my favorite things to do. I was on the swim team starting when I was 4 years old and then on and off throughout grade school. Although I wasn't especially fast. I just loved the water. I used to dream that there was a way I could be like a fish. When I was little, I had a misconception that fish didn't need oxygen. Later on, I learned that, no, most fish have gills that allow them to extract the oxygen, which they need, from the water. So then I just thought it'd be really cool if I had gills. But alas, no gills for Pinky. Oxygen is a really big deal. So many organisms, from fish to plants to humans, need oxygen. And yes, even though plants make oxygen in photosynthesis, they still perform cellular respiration, and therefore plants still need oxygen themselves. There's often a misconception that plants don't need oxygen. That's just not true. So why do these organisms need oxygen? It's similar to why you need oxygen. If you've ever wondered why you need to breathe, which is done by the respiratory system in your body, zoom into the cell level. Cells in your body use the oxygen you inhale to perform cellular respiration. The formula here requires inputs, otherwise known as reactants, to make ATP. And oxygen is one of those reactants in the overall equation that is needed to break glucose down in forming ATP. Why ATP? ATP stands for adenosine triphosphate, and it's action-packed with 3 phosphates. It has the ability to power many cellular processes. Typically, it's coupled to other things that it may be powering. Now, after losing the phosphate, the molecule is ADP, adenosine diphosphate, because it has 2 phosphates. In cellular respiration, though, there are enzymes that can add another phosphate to it and convert it back into ATP again. This particular formula of cellular respiration is aerobic, meaning overall it requires oxygen. It's pretty complex, and we have a video breaking down the steps. But that's not what this video is about. This video is about what happens when there is no oxygen, because cells still need to make their ATP. So what kind of cells can handle the no oxygen thing?
Well, many types of bacteria can, many types of archaea, yeast, which is a fungus that could be helpful, like making your bread rise, your muscle cells can, for a while anyway, and these are just a few examples. Now, these organisms handle the lack of oxygen in different ways. Some organisms, such as some types of bacteria or archaea, can do anaerobic respiration. They can continue to perform glycolysis, Krebs, and the electron transport chain, just like aerobic cellular respiration. But since there is no oxygen to be that final electron acceptor at the end of the electron transport chain, they use something else. Sulfate, for example. These organisms are specifically adapted to be able to use a different electron acceptor in this anaerobic respiration. Another option is the organism may just stick with doing glycolysis, which doesn't require oxygen, and then the addition of some way to get their NAD+ back. We'll talk about what that means in a minute. This process is called fermentation, and that's what we're going to focus on. Fermentation is a way to be able to handle the little to no oxygen issue. It allows for glycolysis to happen and for glycolysis to keep going. That means making ATP where there is no oxygen. And while you won't make as much ATP in this process as you would aerobic cellular respiration, you can't be too picky when oxygen isn't around. Okay, so recall what glycolysis is from our cellular respiration video. In glycolysis, you take glucose, a sugar, and it gets converted into pyruvate. This takes a little ATP cost to actually start it up, but overall, per glucose, you make 2 net ATP molecules, and you also produce 2 NADH. Recall that NADH is a coenzyme and an electron carrier. We also need to mention that NADH didn't just poof appear as a product. No, because NAD+ actually was reduced to NADH when it gained electrons. And if the words reduced and oxidized are confusing, you can remember the famous Leo Gurr mnemonic. Lose electrons oxidized, gained electrons reduced. So NADH to NAD+ is oxidation because it loses electrons, and NAD+ to NADH is reduction because it gains electrons. Now, NADH, the electron carrier, would normally be delivering the electrons gained to the electron transport chain, if this was aerobic cellular respiration. Once losing their electrons, NADH would be oxidized into NAD+ and be ready to be involved all over again in glycolysis. But there's no electron transport chain in this fermentation process, so we've got to regenerate the NAD+ somehow. NAD+ is needed here, after all, for glycolysis to continue. Fermentation, therefore, adds another little step to the end of glycolysis, a step to help regenerate NAD+. This happens because fermentation allows NADH to give its electrons to an electron acceptor, which, in the 2 fermentation examples we are going to give, will either be a derivative of pyruvate or pyruvate itself. So here we go with 2 types of fermentation, which both result in different products from pyruvate. Alcoholic fermentation is done by some types of yeast. So first glycolysis happens, which yields 2 net ATP, 2 pyruvate, and 2 NADH. Now, we need this step to regenerate the NAD+, so we can keep doing glycolysis. The 2-pyruvate is used, which will ultimately produce carbon dioxide and 2-ethanol, alcohol. But the derivative of pyruvate shown here, acetaldehyde, can act as an electron acceptor in this process, so the 2-NADH can be oxidized to 2-NAD+, so that glycolysis can start all over. Since alcohol is a waste product in this process, this is why many yeast are used to produce alcoholic products. Yeast also can do alcoholic fermentation in making bread, and the carbon dioxide product we mentioned is involved with helping the bread rise. The tiny amount of alcohol produced in the short fermenting time of bread will evaporate in the baking process. Lactic acid fermentation is done by cells such as your muscle cells, for example. While your muscle cells can do aerobic cellular respiration, they can shift to lactic acid fermentation if they experience an oxygen debt. This could happen if you're working out very intensely, where your blood is unable to deliver a sufficient amount of oxygen to them for their demand. Just like alcoholic fermentation, we start with glycolysis, that yields the 2 net ATP, 2 pyruvate, and 2 NADH. But now we need the step to regenerate the NAD+, and this step is a bit different from alcoholic fermentation. The 2-pyruvate on the reactant side will ultimately yield 2-lactate. The pyruvate can act as an electron acceptor, allowing NADH to be oxidized to NAD+, so that glycolysis can start all over. By the way, this lactate product, or specifically its other form, lactic acid, has often been blamed for the muscle soreness that occurs after intense exercise. In many of my teaching years, this was often discussed, but there's some recent research that may contradict that. Science is so cool. Check out our further reading suggestions and our video details to learn more. Lactic acid fermentation is also done by bacteria that are involved in making yogurt, and lactic acid can contribute to its sour taste. So overall, fermentation is a pretty remarkable process. Although, it does make us appreciate oxygen because despite how absolutely awesome fermentation may be, it just can't make as much ATP as aerobic cellular respiration. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.